we've spent the last 2 days and this morning talking about the immune epitope database. This is obviously what the workshop is all about. But for the next couple of hours, we'll be introducing new resources. And the next one in particular is very, very exciting. It's something that the IEDB does not cater to. So it does not cater to cancer data by design, or did not previously. And was something that we did feel very passionately about, that it was necessary to have a cancer epitope database. So a couple of years ago, after much lobbying and grant writing, we were awarded by the National Cancer Institute a 5-year grant to create the cancer epitope database and analysis resource. So we call CEDAR, they call this CEDAR, and we call it basically a sibling database to the IEDB. And there's a lot of exciting work going on in this space. And compared to IEDB's almost 20 years, CEDAR is in its early stages, but has a lot of capacity and a lot of growth and a lot of usefulness. And it will continue to grow as well as we continue to expand this resource. So that's what the next 2 presentations will focus on. Shortly, I'll introduce Zeynep to present the CEDAR work holistically and give you an introduction. And then we'll be joined by Alex after that to show you how you can apply CEDAR and its data to research and draw insights and conclusions from the database. So yeah, we're very excited to now introduce Dr. Zeynep Kosaloglu-Yalcin to present the Cancer Epitope Database and Analysis Resource, also called CEDAR. Thanks very much, Zeynep.
Thank you, Nina. Okay. I want to start with a warning that I am unfortunately sick, so I do want to apologize for any coughing or sneezing during the presentation. Yeah, thanks, Nina, for the nice introduction. I will present the work we have been doing for the Cancer Epitope and Database Analysis Resource over the almost past 3 years now. Let me start with a brief overview of the different types of cancer antigens. So starting here on the right side, we have the overexpressed antigens. So these are antigens that are expressed in both the healthy cells down here and the cancer cells, which are illustrated up here. So these antigens are expressed very highly in cancer cells because the cancer has some kind of advantage of expressing them. And in the normal cells, they are expressed in lower levels. So examples of this category are, for example, the HER2/neu genes or the survivin gene. Then here we have the tissue-specific antigens, also called differentiation antigens. These antigens are expressed only in specific tissues and in cancer cells, again, because they have some kind of advantage by expressing them. So, for example, they are only expressed in melanocytes. So this is a prominent example in skin cancer, for example. So they are not significantly expressed in any other normal cell. So they can also be at the desire to be analyzed in specific cancer types that are tissue specific. So examples for this are the MART1 gene or the prostate specific antigen PSA. The next type of antigens we can see here are the tumor specific antigens. They are also called germline antigens because they are expressed during the development of a fetus. But after birth, these antigens are not expressed in normal cells anymore, except for some reproductive cells, which we can see here. But those cells, they do not express HLA, so the peptides from these antigens will never be presented to the immune system. Tumor cells, however, they use demethylation to express them again because they provide some advantage for the tumor growth. So we can see these germline antigens presented on a lot of tumor cells, actually. The most tumor-specific antigens, however, are the mutated antigens. They are also called neoantigens. Tumor cells, they acquire certain mutations in their DNA, which is illustrated here by this green coloring. And if the mutation in the DNA is in a coding region, they can be translated to a mutated protein, and then the mutated peptide can be presented on the cell surface to the circulating immune cells. Because there are no mutations in the normal cells, by definition, these mutations will never be presented on normal cells, which makes these neoantigens highly tumor specific. So all of these different tumor antigens are very important for cancer research. They are important to understand how the tumors develop and how they can be better treated, and to also develop immunotherapies specifically. So the motivation for the CEDAR project, as Nina mentioned, is that we have the IEDB, which is a great database for all things epitope data related, but cancer was never part of the IEDB. So we received the funding from the National Cancer Institute to develop a resource specifically for cancer epitopes. Basically do the same we did for IEDB over the past 20 years, but now specifically for cancer. And just as we did for the IEDB, we developed CEDAR to have two sides. We have the database side, where we curate cancer epitope data from the literature and link them to all the relevant information, the biological, immunological, and clinical context we find in the publications. And we also have the analysis resource, where we provide computational epitope prediction tools and analysis tools specifically designed for a cancer researcher. Okay, so as I said, fortunately, we were able to use a lot of our experience from the IEDB, but we had to also generate specific features so we can capture cancer data more efficiently. To do so, we interviewed a lot of experts from the cancer research field. And then we asked them, what do you need to be captured specifically in such a database that you can do your research efficiently? So what the example is that we introduced for here, as you can see here, we introduced different vaccination types. So in the IEDB, vaccination was only prophylactic. In cancer, we also specifically have therapeutic vaccinations. Another example is that we now also capturing cancer types in more detail. Previously, we would only say skin cancer or colorectal cancer, for example, but now we also added the stages of the cancer to the database. So once our database was established, we started curating the cancer epitope data. And we used the same curation process we use for the IEDB, which is outlined here. So how this process starts is PubMed is automatically queried every 2 weeks to retrieve any papers that might contain epitope data. And we have an automated classifier that groups papers into categories such as infectious disease, allergy, or cancer. As a next step, this classification and also the papers themselves are manually reviewed to see if the data is really usable and if it should be curated. And if the paper is deemed curatable, it will be assigned to a curator to go through the paper and extract the data and put it into CEDAR. So as you can see here, of all the papers we have identified as containing epitope data, 13% of those papers are related to cancer. And here in this table, we have a breakdown of what has been curated so far. Prostate cancer and neoepitope papers are the two categories we started with, actually. So these are almost complete. I have to mention, though, we will never fully reach 100% probably, because new papers are being published and then they're being added to the curation queue. So after we curated these two categories, we switched to a year-based curation queue. So we started from this year, and then we're working our way back to curate papers from previous years. And this graph down here shows how many more papers might be curated for each date range. And as you can see, we still have thousands of papers that might contain epitope data and will eventually end up in CEDAR. So before I go a bit more into detail about the data we have curated so far, I want to show you how the website looks currently. So as you can see, it is very similar to the IEDB site, but we have some important distinctions. So I guess the Epitope source panel had the most change compared to the IEDB. We added these checkboxes here. So as I've mentioned at the beginning, we have different types of cancer antigens, and researchers are oftentimes have the niches and they're interested in specific types. And using these checkboxes, they can specifically search for them. We also added this checkbox here, which says other antigens from the same reference. We had to add this option because oftentimes a paper will contain epitope data from cancer, but then also some data, for example, from COVID patients. Then the whole paper is curated as part of CEDAR, but by deselecting this checkbox, you won't have the non-cancer epitopes in your query results. I guess host, assay, and image restriction mainly stayed the same. And we have introduced this panel cancer down here. So here you can choose, we added a disease finder. When you start typing your cancer type of interest, you can select the cancer, but I will show you this later in a query example also. You can also select the stage that you are interested in, like stage 1, stage 4, and so on. And down here we have the radio boxes to choose if you are interested in naturally occurring diseases or in a animal model. And we also added this radio box for vaccination, so that the user can search for epitope data from specifically from hosts that were vaccinated before with a cancer vaccine. So here we have a brief overview of the data we have currently in CEDAR. We have been curating data for almost 3 years now, and I think it's impressive how much data our curation team was able to curate in this short time frame. As you can see, we have much more ligand elution assays than T-cell or B-cell assays, but that's because mainly these assays are high throughput and they generate thousands of results with one experiment. We have also currently paused the curation of ligand elution assays to focus more on T-cell and B-cell assays. When we have a look at the T-cell data we have in CEDAR right now, we can see that most epitopes are from common cancer types. Here we have the top 10 disease types in terms of how many epitopes have been curated. As you can see, we have melanoma here, we have, which is skin cancer, we have colorectal cancer and lung cancer, and these are all cancer types that occur most frequently in the population, so they are very heavily studied. As you might have noticed, we also have the healthy here. That's not a mistake, because in a lot of experimental settings, cancer samples are not available, and researchers often use a healthy sample to test their cancer epitopes on. Okay, and here we have the top 10 antigens that we have in CEDAR right now, and again, we can see MHA10 and MLA-NA, which are both melanoma antigens, which makes sense, again, because melanoma was the most studied one here. We also have TP53 and KRAS as the top 2 and 3 antigens. These are actually antigens that are most frequently mutated, and they produce new antigens. Okay, speaking of neoantigens, we actually have a special interest in neoantigens right now. We, and also in the cancer community in general, neoantigens are very prominent because they are being used for developing cancer immunotherapies, such as therapeutic vaccines. In CEDAR right now, we have almost 22,000 neoantigens curated. Most of these have been tested with a T-cell assay, but were not recognized by the T-cell. But we also have around 3,400 neoantigens with positive outcomes. So this is a good amount of data we have here together, and that we can do some statistical analysis on. And this is actually a project we are currently working on to understand what makes a neoantigen being recognized by T cells, and what are specific features that might be important to consider in future analysis. So I want to show you now some example queries. The first example I want to show is how to search for neoantigens in lung cancer. Here we use here the checkboxes. We deselect all of them and just keep the neoantigen box selected. For this example, we are interested only in human and in T-cell assays and in class 1 restricted epitopes. And as I mentioned earlier here, we started typing lung, and then we get the disease tree that we have in CEDAR, and then you can select the lung cancer here. As a result, you get results similar that you know from the IEDB. We have the current filters top here, and then you have the different tabs you can click through and look at the data. So this is the Epitope tab, where you can see all the different epitopes that have been reported in lung cancer. And because we specifically search for neoantigens, as you can see here, the antigens always have the prefix neoantigen. And this is because neoantigens are produced by mutations. So the antigen itself is also slightly different from the wild type antigen. So this is a mutated epitope. So if you want to know what the wild type epitope, the corresponding wild type epitope was, you can click here on the epitope ID, which brings us to the epitope summary page. And then here it is mentioned what the wild type peptide of the epitope was. Okay, as a second example, which will be relevant for Alex's talk next, I want to show how to query for prostate-specific antigens. Here we worked with the antigen finder in the epitope source panel. We start typing prostate, and then we get the option to select prostate-specific antigen, which we select. And then again, similar, we select human, T-cell. This time we're interested in both positive and negative outcomes. And we have MHC class 1 restricted epitopes. And here we're interested in any cancer type, but only in cancers that occurred naturally in the patient. And this is the result we get. So only one antigen, obviously, because we filtered for the prostate-specific antigen. And then we have 21 unique epitopes from this antigen, which were described with 133 essays, and 23 references have reported on them. And if you want to further refine or change the query, you can also always use this left panel to change the search and update your results. So CEDAR, as you can see, is completely functional right now. But we are also working on adding additional features that will be helpful for cancer researchers. Again, these features are mostly related to neoantigens, because neoantigens are very specific for cancer, and we did not have anything like that in the IEDB, so we have to do a lot of changes to accommodate them in CEDAR. So currently, you can only perform a general query for neoantigens, as I showed before, but oftentimes researchers are interested in neoantigens produced from specific mutations. So we are currently working on adding this additional search field here, where you can type a specific mutation for the specific antigen you're interested in. So in this case, for example, you would be interested in the antigen epitopes from KRAS that have the mutation from G to D at position 12. Another feature we are working on is related to mutations again. So CEDAR is a peptide-based database. But as I've shown in the beginning, neoantigen is first caused by a DNA mutation. And knowing more details about the DNA mutation can be helpful for cancer researchers. So we're currently working on a feature to provide information about the mutation underlying the neoantigen. To achieve this, we are collaborating with external tools, actually. So first, we have a tool from UCSC called Transvar, which can take the peptide and map it back to the genome, which means you will know, so you can translate your amino acid mutation back to a DNA mutation. And once we have the DNA mutation information, we can use this tool OpenCravat from Johns Hopkins. OpenCravat is an annotation tool that is linked to, I guess, hundreds of different tools and databases, and we can use open cravat to just retrieve, to put in our DNA information for the peptide, and then get all the additional information we need about the mutation. So this is a mock-up of how we envision the epitope summary page to expand. So it can include all those additional information. So these are all would be new fields that we are currently working on adding. So so far, because we are a peptide-based database, we only have like protein names and protein identifiers, but we also want to add the gene symbol because of the DNA mutation link nature of the cancer data. We want to add the specific details about the protein change. You will still have the UniProt accession, the protein ID, the mutation type, the DNA mutation information, and then also information from these external databases, like how frequent is the mutation in a general population, if it was predicted to be damaging for the protein function. So these are all options that we will be able to get from the Open Cravat tool. And we will also provide a link out to Open Cravat where much more details will be provided for the user if they're really interested to dig into those details. So next I want to talk about the analysis resource. As a reminder, here we want to provide epitope prediction and analysis tools specifically designed for cancer. So we had 4 overarching goals for the analysis resource for CEDAR. So namely, provide prediction tools tailored to the needs of cancer immunologists, develop novel prediction tools for cancer epitopes, provide web implementations for published but hard to access cancer epitope related tools, and use curated cancer epitope data to benchmark epitope prediction tools. So for the first goal of developing novel prediction tools, we actually have recently published 2 tools, namely XLF, which is an epitope prediction tool that combines antigen expression and MHC binding, and was shown to perform better in predicting epitopes that are presented on MHC and recognized by T cells. Related to that, we also published the tool PepX, which allows the user to input a peptide sequence, and PepX then will find the source protein for that peptide, and then provide expression levels, which can be retrieved from RNA-seq data from public databases, such as the Cancer Genome Atlas, TCGA, or the Cancer Cell Line Encyclopedia, CCLE. So these 2 tools are also highlighted on the CEDAR homepage here at the site if you want to try them out. We also continue to work on developing novel tools. So there's many more features that can be relevant when predicting cancer epitopes. So far we had experimented with the different expression values, but there's other features that can be looked at. For example, the clonality of the mutation, or in case of neoantigens, we can see how similar the neoepitope is to the self-peptides or also to other pathogens. So yeah, we're always working on developing, researching these new features and developing novel tools. For the second goal of providing tools tailored to the needs of cancer immunologists, we also have 2 tools currently in the pipeline. So the first tool that we worked on is what neoepitopes are generated by a given mutation, by a given DNA mutation. So as we know, the DNA mutation occurs in the DNA and then is translated to a mutated protein, and which can generate mutated peptides. So in a practical sense, what the user always will have is the DNA mutations in this VCF format, which is basically a table which details the position of the mutation in the chromosome, and then what nucleotide was the mutated to which nucleotide. And then we actually have been using an internal tool already for a long time, which takes this VCF file with the DNA mutations, and it generates a table with the possible mutated peptides. And yeah, this will also be made available now to the public as well as part of the CEDAR resource. Another detail that users often requested is to perform side-by-side predictions for mutant and wild-type peptides. As we know, we have a new antigen, which is a mutated peptide, but we also have the wild-type counterpart for the peptide. So far, what many people do is they first run all the different predictions for the mutated peptides, and then they run the predictions for the wild-type peptides, and then they use complicated custom scripts to bring it all back together. But we want to make this much easier, where the user can input the paired wild-type mutant peptides, and then we will provide the side-by-side results with the affinity predictions, for example, for the wild-type and the mutant, and also the difference between the two, which researchers are really interested in. So these two functions, together with the PepX tool, will actually be part of one of the next-gen tools. I think this was mentioned already yesterday. We are planning to do this in March next year. So keep an eye out of that if you want to use it, these tools and functionalities. So our third goal for the CEDAR analysis resource was to provide web implementations for published but hard to access cancer epitope related tools. So we did do a lot of research. There's a lot of papers out there that publish tools that claim that the tool is the best. So we have a list of tools that might be interesting, and we are working on prioritizing these tools based on community need and based on a cost-benefit analysis. So this is also something that is ongoing. Yeah. This is also something we are actively working on to use the curated cancer epitope datasets to perform benchmarks for prediction tools. We have our epitope data from CEDAR, and anyone who worked with IEDB or CEDAR before knows that we have a lot of details in there. And bioinformaticians oftentimes do not have the biological background to understand all the experimental details and how they might need to filter the data to make it usable for their application. So we are working on providing, so to say, cleaned up versions of these data sets that we can provide to bioinformaticians, and they can easily just take them and just plug it into their machine learning frameworks, for example, and use them to develop new tools or test new tools. And in parallel, we are also using these tools to perform benchmarks. Right now, we are working on performing them manually, but the ultimate goal is to do an automated benchmark like we are doing in IEDB right now. So examples for benchmark targets would be what peptides in the tumor samples are processed and presented on MHC, or what neoepitopes are recognized by T cells from a cancer patient. So these two questions, for example, they might sound very similar, but they are slightly distinct, and it's an important distinction. And we need to know how to use and filter the data based on the experimental details so you can efficiently answer these questions. So if you are interested to learn more about CEDAR and all the cancer functionalities, we have been attending AACR for the past 2 years, and we will be attending CEDAR again, AACR again this year. This is the American Association for Cancer Research. This is the annual meeting, which happens in San Diego this year in April, I think. So we will be booth 3753. So if you're attending the conference, please stop by our booth. And whenever you use CEDAR, or also if you want to learn more about CEDAR, you can cite our paper here, which we published last year in the NAR database issue of last year. And yeah, so this was my last slide. I have a short summary. The CEDAR database is up and running. We are working on adding new features, but you can already try and use the data. We have already curated thousands of data points, and we are moving rapidly. We already have some tools ready, which will be also part of the next gen tools releases early next year. And yeah, we have a good community engagement from conferences and different talks. Yeah, so that's all what I wanted to show. I'm happy to take any questions in the Q&A.
Awesome. Thank you so much, Zeynep. And a special thank you for pushing through for 30 minutes, quite unwell. So obviously Zeynep had hoped she was going to feel better by today, but got worse overnight, but continued with the presentation. So super appreciated. It is.
Thanks for bearing with me.
No, you did a great job. So thank you for that.